Hello, and welcome to your Articulate file for Unit 4, an introduction to the renal system, and this is covered in Chapter 26 of Guyton and Hall. First, I'd like to start with a basic overview of the kidney anatomy. The kidney is about the size of a bar of soap. It is located between the vertebrae of T12 through L3. If you'd like to palpate that, first find your last rib and follow it up superiorly until it reaches the T12 vertebrae. This would be the superior border of your kidney. The right kidney is displaced inferiorly due to the liver. Both kidneys are retroperitoneal, which means they are behind the parietal peritoneum. The kidneys each have an outer cortex layer and an inner medullary layer. The medullary layer has renal pyramids, each with an apex that leads to a minor calyx. Minor calyces merge to form a major calyx, and major calyces merge to form the renal pelvis. Urine, excreted solutes in obligatory water, is transported through these structures to the renal pelvis. The renal pelvis leaves the kidney and turns into the ureter. The ureter carries urine to the bladder, where it is stored until socially acceptable to void the contents. The objective of this unit is to review the anatomy of the kidney, to understand the blood flow into and out of the kidney, to understand how the blood is filtered by the nephron, to understand how much of the filtrate in the nephron is reabsorbed, to understand that the kidneys can also secrete solutes into the nephron that were not filtered, and lastly, to understand how the kidneys depend on renal blood flow in order to filter the blood. The amount of blood plasma that is filtered is called the glomerular filtration rate, or GFR, and understanding how this is maintained by the kidneys is extremely important. The kidneys perform many important jobs for the body. They maintain volume and composition of your bodily fluids and do this through three mechanisms: 1. Filtration, 2. Reabsorption, and 3. Secretion. I will be talking about these three mechanisms here during this Articulate lecture, but we'll also talk about them in more detail during our face-to-face lectures. The kidneys are also an endocrine gland. I'm sure you remember that they are the main source of erythropoietin. You remember that EPO stimulates the bone marrow cells to produce more red blood cells. In this unit, you will also learn about renin, a hormone released by juxtaglomerular cells, also known as granular cells, of the juxtaglomerular apparatus. The kidneys are also very important in ridding your body of non-volatile acids and bases. This means that they have the arduous task of excreting acids and bases that are not in a gas form. Acids that are in a gas form are removed from the body by the lungs. Yes, the kidneys and lungs together do an excellent job of maintaining your acid-base balance. Next, the kidneys can, under severe conditions, help make new glucose molecules out of basic materials like amino acids and fats. This process is called gluconeogenesis, and it helps to maintain blood glucose levels. The liver is the organ primarily responsible for this event, but the kidneys can also perform this function. The kidneys, through their reabsorption processes, reclaim many of the electrolytes that are filtered. Were it not for the kidneys reabsorbing these electrolytes, they would be excreted in the urine. You remember that an electrolyte imbalance like hypo or hyperkalemia, hypo or hypercalcemia, or even hypo and hypernatremia can impair normal cardiac rhythms. Lastly, during this unit, I will discuss how the kidneys maintain long-term control over mean arterial pressure. They do this, of course, by regulating how much urine is produced. Urine is basically a derivative of blood plasma. If more urine is produced, think of this as more blood plasma being lost. If less urine is produced, think of this as more blood plasma being conserved. With more blood volume comes more blood hydrostatic pressure. Remember, the pressure of blood on the vessel walls. With more blood pressure comes more blood flow. With more blood flow, the organs and tissues of the body are well perfused. Let me review some basic anatomy of the kidney. First, the picture shown at the bottom is a nephron, the functional unit of the kidney, surrounded by a capillary bed. A nephron consists of the renal corpuscle and the uriniferous tubule. A renal corpuscle consists of the glomerulus and the glomerular capsule, or Bowman's capsule. A glomerulus is a fenestrated capillary bed. The uriniferous tubules consist of the proximal convoluted tubule, or PCT, the loop of Henle. There is a descending limb and an ascending limb. The loop of Henle leads to the distal convoluted tubule, or DCT. The DCT has two functional parts to it, the early and late segment. Lastly, we reach the collecting tubules and collecting ducts and medullary and papillary ducts. These last features are not part of the nephron, but attached to many different nephrons, and we still need to appreciate them and their physiology. The nephron is called the functional unit of the kidney because this is the structure that will perform all those functions I discussed on slide 2. I will elaborate on all the structures of the nephron throughout this lecture, and I will also elaborate on their physiology both here briefly and in a lecture during our class time called Renal Physiology. The picture on the left shows two different types of nephrons. The smaller one is called a cortical nephron. It is the most abundant type of nephron, consisting of 80% of all the nephrons. It is called a cortical nephron because the majority of this nephron is found in the cortex of the kidney. The name of the capillary bed that surrounds this type of nephron is called a peritubular capillary bed. In contrast, 20% of all the nephrons in the kidney are a type called juxtamedullary. They are called this because their loop of Henle extends deep into the medullary region. Juxta means next to. The juxtamedullary nephrons are extremely important in creating a scant, highly concentrated urine. They establish a countercurrent exchange system, which I will address at a later date. The name of the capillary bed around this type of nephron is called vasa recta, which translates to mean straight vessels, and they too extend deep into the medulla, and these vessels will absorb any excess salt or water in the medulla. The primary goal right now is to understand how blood flows into and out of the kidney. Blood is ejected out of the left ventricle into the aorta. This blood flows through the aortic arch to the thoracic aorta to the abdominal aorta. The first large branches off the abdominal aorta are the celiac trunk, supplying blood to the stomach, liver, and spleen. Next is the superior mesenteric artery, followed by the suprarenal arteries. Finally, blood enters the renal arteries and now starts the journey of blood throughout the kidney. The renal arteries lead to segmental and lobar arteries. Lobar arteries branch into vessels that travel in between the renal pyramids, and these are called interlobar arteries. Now these vessels arch over the base of the renal pyramids and turn into the arcuate arteries. The arcuate arteries branch off smaller vessels that extend into the cortex, and these tiny branches are called interlobular arteries, also known as cortical radiants. Now I really need you to listen. Afferent means incoming, and efferent means outgoing in anatomy. The afferent arteriole is bringing blood into the glomerulus that is tucked inside Bowman's capsule. Again, the glomerulus is a special type of capillary bed. It is a fenestrated capillary. This means it is very porous and has high permeability. Traditionally, a vein would follow after a capillary bed, but in the kidneys there is an arterial portal system. This means, following the glomerulus, there is another arteriole called the efferent arteriole, which means it is leaving from the glomerulus. Now, last unit, you learned about how capillary beds release local factors to control the blood flow through them. You also learned that blood flow can be controlled by regulating the radius and hence resistance of the arteriole leading to the capillary bed. It will be no different for the kidney, except now there is an arteriole upstream of the capillary bed and downstream of the capillary bed, and both these arterioles can change their radius and create more or less resistance to blood flow. Ask yourself these questions. If I constricted the afferent arteriole, what would blood flow be like into the glomerulus? What if I vasodilated the afferent arteriole? What if I vasoconstricted the efferent arteriole? What would happen to blood flow in the glomerulus? What if the efferent arteriole was vasodilated? Did you answer reduced blood flow, increased blood flow would be dammed up in glomerulus, and less blood dammed up in the glomerulus, respectively? If you did, well done. Let me now finish how the blood flows through the efferent arteriole into a second capillary bed. Again, the name of this capillary bed is called the peritubular capillary bed when it surrounds a cortical nephron, and it is called vasa recta when surrounding a juxtamedullary nephron. The solutes and water reabsorbed by the nephron will be placed into the interstitial space, and then the solutes and water will flow through bulk flow into this second capillary bed. The blood then flows into an interlobular vein, to an arcuate vein, to an interlobar vein, which then converges into the renal vein, back to the inferior vena cava, into the right atrium. There are no lobar nor segmented veins. I bet that bothered you in anatomy, huh? Take a really good look at this drawing of a nephron. It shows the nephron without the peritubular capillary bed or vasa recta, but it does show the afferent and efferent arterioles and glomerulus. Blood is going to enter the glomerulus via the afferent arteriole. Within the glomerulus, there are Starling forces that will favor the movement of water and dissolve solutes out of the glomerulus and into Bowman's capsule. The fluid in Bowman's capsule is called filtrate to reflect that it is a derivative of plasma, but not exactly like plasma. The water with dissolved solutes, or filtrate, now enters the proximal convoluted tubule. The filtrate in the PCT is most like plasma, but its consistency will be changed as it flows through the nephron. In the PCT, most of the water and solutes will be reabsorbed. The remaining filtrate will then proceed to the descending limb of the loop of Henle and then to the ascending limb. There is a thin segment and a thick segment. Next comes the distal convoluted tubule, and there is an early and a late segment. I want you to look here at the arrow and notice how the early distal convoluted tubule is found in between the afferent and efferent arteriole. This is an important interaction. These structures are in a functional contact with each other to create the juxtaglomerular apparatus. I'll be discussing the physiology of this structure in this lecture and other lectures in this unit. After the distal convoluted tubules, the filtrate now enters the various collecting tubules and ducts. These structures can still reabsorb solutes and water, but only under hormonal control. The filtrate by this time is most like urine. In the next few slides, I will go through each of the nephron segments and give you an overview of their function. Let's begin with the glomerulus tucked within Bowman's capsule. Remember that the glomerulus is a fenestrated capillary bed. This means that it is highly permeable and porous. Pretend you are traveling in whole blood and have just arrived in the glomerulus and look out through the fenestrations within the endothelial cells. On the other side of the endothelium, you would see a sheet of proteins called the basement membrane. These aren't plain proteins. No, these have sugar molecules attached to their amino acids, and this makes them proteoglycans. After the basement membrane, you would now see a layer of support cells called podocytes. They wrap themselves around the glomerulus to give it support, and extensions of their cell membrane makes them look like they have little feet, and these extensions are called pedicels. The pedicels interdigitate to create filtration slits. Now, as you are looking up through the fenestrations, notice some of the plasma moving through the fenestrations and filtration slits into Bowman's capsule. But also notice that the proteins stay behind. The fenestrations are plenty big enough to allow the plasma proteins through, yet even the smallest proteins stay in the glomerulus. Why is this? The plasma proteins and the proteoglycans of the basement membrane are all negatively charged. So, size has nothing to do with the proteins not getting through the fenestrations. Rather, it is because of the charges repelling each other. In fact, I'd like to share with you the experiment that proved this. The graph on this slide summarizes the results from an interesting experiment that proved that plasma proteins did not get through the filtration membrane due to charge repulsion. First, the average size in diameter of the fenestration is about 700 angstroms. Most plasma proteins are 20 to 42 angstroms in diameter. So, the fenestrations are plenty big enough to allow plasma proteins to enter Bowman's capsule, yet they don't. In the experiment, the researchers took dextran. It is a polysaccharide. The researchers took different sizes of this polysaccharide and also attached molecules to it to give it an overall negative charge, shown in black, or neutral charge, blue line, or positive charge, shown in red. Notice that the neutral or positively charged dextran molecules could move through the filtration membrane even when their molecular radius was rather large. Notice the x-axis. But notice the negatively charged dextran had a very difficult time moving through the filtration membrane even when the size of the dextran was very small. So size of the plasma proteins isn't the limiting factor that prohibits their filtration. Rather, it is the negative charges of the plasma proteins and the proteoglycans of the basement membrane that repel each other. In fact, our filtration membrane loses some of its charged proteoglycans as we age. Thus, some plasma proteins do begin to leak out of the glomerulus into Bowman's capsule, and protein ends up in our urine. Protein in your urine should not be present, and if it is, it usually reflects damage to the filtration membrane. The proximal convoluted tubule is made up of simple cuboidal cells. On their apical side, the side that faces the lumen of the nephron, these cells have many microvilli. This means increased surface area. Why would these cells need increased surface area? They need increased surface area in order to insert integral proteins. These integral proteins are going to engage in secondary active transport, and you will learn about this, again, during lecture. These cells also have a tremendous amount of mitochondria found on their basolateral sides, the sides facing away from the lumen. These cells are working all the time. They require huge amounts of ATP, and thus they are loaded with mitochondria. Their basal side, that's the side that faces the interstitial space, is also highly invaginated. Why? To increase surface area. Again, the increased surface area is needed for integral proteins. These integral proteins will be engaged in primary active transport. The loop of Henle has several parts to it: the descending limb, the hairpin turn, the thin ascending limb, and the thick ascending limb. Functionally, the descending limb, hairpin turn, and thin ascending limb have similar physiologies. From the PCT, the epithelium changes from simple cuboidal to simple squamous in the descending limb, hairpin turn, and thin ascending limb, back to simple cuboidal in the thick ascending limb. The descending limb, hairpin turn, and thin ascending limb are only permeable to water. That means water moving out. So the solutes won't be reabsorbed, only water. Why? First ask yourself, how do solutes move across a membrane in the first place? Solutes are usually charged and thus need a carrier of some sort. And the region of the nephron that consists of simple squamous cells simply do not express these carrier proteins. They don't turn these genes on. That's the reason why they are impermeable to solutes leaving the nephron. The hairpin turn and thin ascending limb behave the same way. On the thick ascending limb, now the cells are impermeable to water, and they only move solutes. The question is why? They express carriers for solutes but do not express the genes to create a water pore called an aquaporin. Thus, they will not be permeable to water. This area creates a rare event that may be foreign to your thinking. Solutes will move and water will not follow. More importantly, the loop of Henle with a setup on the descending limb to reabsorb water and the solutes staying inside, and the thick ascending limb with the solutes leaving, but now water is trapped inside, creates a countercurrent multiplier exchange system. Filtrate flowing down the descending limb becomes ultra concentrated, and filtrate moving through the thick ascending limb becomes increasingly dilute. The two feed into each other, and I will discuss this in more detail in lecture. The distal convoluted tubule is made up of simple cuboidal cells, and on their basal side, again, they have a lot of invaginations for the insertion of proteins. These proteins will engage in primary active transport. On their apical side, the side that is facing the lumen on the nephron, we don't see microvilli like we did in the PCT. But they still have a decent amount of mitochondria because they need ATP in order to power some of these integral carrier proteins engaged in primary active transport. The distal convoluted tubule has two parts, an early and a late segment. The early distal convoluted tubule is in a functional contact with the afferent and efferent arteriole and creates the juxtaglomerular apparatus, which I will discuss later in this lecture. The late distal convoluted tubule and everything else after that, collecting tubules, collecting ducts, all of those areas are now considered to be under hormonal control. In other words, from the late distal convoluted tubule onward, these structures will reabsorb more sodium chloride and water under hormonal direction. If you need more water reabsorbed, these structures are ones that will obey hormones that will tell them to become more permeable to water. Or if you need more sodium chloride, these structures will absorb more. And again, you're going to learn about that during lecture. As I said in the previous slide, all structures from the late distal convoluted tubule onward, collecting tubules, collecting ducts, medullary ducts, etc., will be under hormonal control. Again, these structures can reabsorb your sodium chloride and water reserves. They can reabsorb more sodium chloride and or water as long as a hormone is around that tells them to do so. The hormones that direct this activity are aldosterone and antidiuretic hormone, or ADH, and I will be discussing them in more detail throughout this unit. This slide is so important as it reinforces Ohm's law with respect to the kidney. Remember, flow equals change in pressure over resistance. In this unit, I am using flow to reflect renal blood flow. That means the amount of blood that is entering and leaving the kidney. A change in pressure is the hydrostatic pressure of the renal artery minus the hydrostatic pressure in the renal vein. That's the change in pressure. That change in pressure is what pushes the blood through all the vessels that we just went over in slide 5. Resistance is going to be the sum of all the resistances in all of those renal blood vessels. The vessels that can afford the greatest resistance are the afferent and efferent arteriole. It is these two vessels that will cause the most dramatic effects in renal resistance, and they will also regulate the amount of blood that enters and exits the glomerulus. For Unit 3, you learned that cardiac output was 5 liters per minute, and this represented flow. Some of this blood did flow to the kidneys. The amount of blood that entered the kidneys is called renal blood flow and is roughly 25% of cardiac output. This means that roughly 1,250 milliliters of the 5,000 milliliters goes straight to the kidney every minute. That is a huge amount of blood flow. Now, remember when we pretended to be in the glomerulus, and I said that red blood cells don't get through the filtration membrane, but plasma does. So plasma is 55% of whole blood, right? So if renal blood flow is 1,250 milliliters per minute, but only plasma with its dissolved solutes can move through the filtration membrane, then only 660 milliliters per minute can be filtered. 1,250 milliliters per minute times 0.55 is 660. When this 660 milliliters of plasma is in the glomeruli of the kidneys, do you think that the entire 660 milliliters per minute is filtered through the glomerulus and enters Bowman's capsule? That can't happen, because then how would the red blood cells move out of the glomerulus? So, of the 660 milliliters per minute of plasma that arrives per minute into the glomerulus, about 20% of that plasma enters Bowman's capsule. And that 20% means that roughly 125 milliliters of your plasma is being filtered. That is called glomerular filtration rate. It means it's the amount of plasma per minute that leaves the glomerulus and enters Bowman's capsule. Of that 125 milliliters per minute, about 123 milliliters per minute is reabsorbed by the nephron and goes back to your bloodstream. That means about 2 milliliters per minute of urine is dripping into your bladder. In other words, most of the plasma that is filtered is reabsorbed. Very little gets to the bladder as urine. Maintaining GFR is very important. You need massive amounts of plasma to be filtered rapidly because toxins like urea, uric acid, and creatinine are very bad for the body. These substances are wastes and can cause disruption of normal physiology and tissues. Urea is a product from protein metabolism. Uric acid results from nucleic acid metabolism, and creatinine is a byproduct of skeletal muscle metabolism. The kidneys want to filter these wastes out very quickly, but reabsorb the solutes that the body does need. Let's review those concepts from the previous slide again. The kidney is responsible for many functions, but 3 of the biggest include filtering your blood, reabsorbing most of the filtrate, and secreting waste that did not get filtered. Let's review the volumes of blood and filtrate that are occurring every minute. First, renal blood flow, or RBF, is 25% of cardiac output. 5 liters per minute times 0.25 equals 1,250 milliliters. I know throughout most of the semester I have not been too big on numbers, but this is important. You need to know these numbers. Of that 1,250 milliliters, only 55% of that volume is the part that could be filtered through the filtration membrane. That means that renal plasma flow is 660 milliliters per minute. Of this 660 milliliters that arrives at the glomeruli, of all the renal corpuscles, about 20% of that actually gets through the filtration membrane. This is called glomerular filtration rate, which is 125 milliliters per minute. The filtration fraction, 20%, reflects the amount of plasma that is filtered versus the amount that arrived in the glomerulus. 125 divided by 660 is 20%. As the filtrate now goes through the uriniferous tubules, we have another process occurring, reabsorption. The cuboidal and squamosal cells are reabsorbing the solutes the body needs to maintain and placing them in the interstitial space. When the solutes are reabsorbed, water passively flows and follows them. Further on in the uriniferous tubules, the cuboidal cells can also do a third function for your body. Anything that did not get through the filtration membrane can be actively removed from the peritubular capillary bed and placed into the lumen of the nephron in order to be urinated out of the body. Now, those three functions, filtration, reabsorption, and secretion, are all a part of an equation for excretion. Excretion means fluids leaving our body. Excretion means whatever the nephrons filter, plus whatever the nephrons secrete, minus whatever the nephrons reabsorb. This is an important equation and one we will talk about more in later lectures.